All right, let's dive straight into this explainer. Because right now, literally at this very second, there is a massive hidden war raging inside your body. Millions of microscopic invaders are relentlessly trying to breach your cellular systems, and your body, well, it's fighting back in some truly miraculous ways. Today, we're breaking down the whole shebang, your multi-layered defense systems, specifically intrinsic and innate immunity.
To kick things off, we really have to ask ourselves, what exactly goes down the very moment a virus tries to invade? See, we usually think about immunity in terms of antibodies or vaccines, right? Things that take days, maybe even weeks to actually develop. But here's the catch. A virus replicates incredibly fast. I mean, blisteringly fast. If your body waited days to respond to an infection, the war would be totally lost before your immune system even realized it was fighting. So we need to zoom in on the absolute front lines, those absolutely crucial first few minutes.
It helps to think of a viral infection almost like a medieval siege. Before a virus can even touch a cell, it has to get past your body's perimeter castle walls. We're talking continuous physical and chemical barriers. This is your respiratory mucus, your saliva, that harsh stomach acid, your tears, your skin, and chemical defenses known as defensins. These are your always-on, 24/7 perimeter walls.
But if those outer walls fall, your host defense kicks in, and it follows a really strict chronological timeline. Right past those continuous physical barriers, a virus immediately slams into the intrinsic immune system. Bam! Immediate response. If that fails, within minutes to a few hours, the broader innate immune system mobilizes, bringing in heavy hitters like natural killer cells and neutrophils. And actually, it's only after hours or even days that the adaptive immune system, your T cells and B cells, finally enter the battlefield.
Notice how the massive swarm of viruses at the start gets severely thinned out as they're forced to cross these successive barriers. It's an intense gauntlet. The vast majority of invaders, they never even survive the anatomical barriers. Those that do make it through are immediately faced with intrinsic cellular defenses. It's only the absolute most resilient, hardcore viruses that survive long enough to trigger that acquired adaptive immune response.
Now, it is super easy to confuse intrinsic and innate immunity. But try thinking of it like this. Intrinsic immunity is a sprinter already in the starting blocks. It relies on systems that are already present in the cell, so it's ready to go instantly, zero delay. Innate immunity, on the other hand, is more like a runner who's still stretching. It requires a brief warm-up period of minutes to hours to actually mobilize specialized immune cells to the site of the infection.
So the million-dollar question here is, how do our cells actually spot the enemy? Well, they do it by detecting PAMPs. You can think of these as the unique molecular fingerprints of the intruders. Pathogen-associated molecular patterns are structures that viruses and bacteria have, but that our own cells definitely do not. It could be a specific type of double-stranded RNA, or maybe some specific DNA motifs. Basically, our cells use these unique features to positively identify that, hey, something foreign and dangerous has entered the area. And to read those molecular fingerprints, our cells use PRRs. These act as your cellular security guards, designed specifically to recognize those viral fingerprints. Pattern recognition receptors are specialized proteins hanging out on our cell membranes and even inside our cells. The moment a PRR binds to a PAMP, boom, the molecular tripwire is triggered and the cell knows without a doubt it is time to fight.
But that brings us to the next logical question, right? How does a cell actually sense a virus once it has already breached the outer membrane? Because viruses, they don't just politely stay on the outside of the cell. Their entire goal, their only goal really, is to sneak inside, totally hijack the cellular machinery, and start replicating like crazy. So our cells desperately need a way to detect the intruders that are actively hiding in the cytoplasm.
And here is how they do it. Even once inside, the virus is still being hunted. Specific cytoplasmic receptors act as this incredible internal security system. You've got toll-like receptors designed to detect foreign RNA and other sensors like CJs that spot foreign DNA. It doesn't even matter which internal alarm is tripped. They all trigger a massive, complex signaling cascade downward, straight to the nucleus, to completely change the cell's gene expression.
Okay, so once the virus is sensed and those genes are expressed, the cell unleashes cytokines and interferons. Think of these as the ultimate SOS flare signals going up to warn the rest of the body. Cytokines are small proteins that act as messengers to attract and stimulate your immune defense cells. And interferons? They are a very specific, highly potent class of cytokines whose main job is to activate these intense, scorched earth antiviral programs in the surrounding tissue.
Let's look at how this builds because it's fascinating.
The interferon spreads out to neighboring cells, binding to their receptors and flipping the switch on interferon-stimulated genes, or ISGs. This creates a highly fortified antiviral state that basically halts viral reproduction in its tracks. Picture this: an infected cell literally sacrifices itself by pumping out interferon. The healthy cells next door receive this urgent signal and immediately start producing hundreds of different ISGs. This creates a local environment where it becomes practically impossible for the virus to replicate, assemble, or spread any further.
And one of the absolutely most amazing antiviral programs deployed by these interferon-stimulated genes involves just 3 simple steps, ending with a protein called BST2, also known as tetherin. Tetherin basically acts as molecular superglue. When the warning signal is received, tetherin rushes to the cell membrane with one singular job: completely deny the virus an exit strategy.
It literally traps dangerous enveloped viruses like HIV-1 and SARS-CoV-2. Tetherin essentially embeds one anchor into the host cell membrane and the other directly into the newly formed virus. The virus thinks it's about to float away and infect a new cell, but instead it finds itself chained to the host, entirely neutralized. It's just a brilliant defense mechanism. But seeing all this incredible firepower and how effectively it shuts down major viruses, we have to stop and ask, why should the interferon response be carefully regulated? Why not just leave it on forever? I mean, if interferons are this incredibly good at stopping viruses, wouldn't we want them circulating all the time to keep us perfectly safe?
Well, the answer to that connects directly to how you feel when you're sick. That massive cytokine signaling? That's exactly what causes the fever, the fatigue, the total lethargy you experience when your body is fighting off a bug. When your body is just pumping out interferons and other inflammatory cytokines, it signals your brain to crank up your body temperature. It triggers your liver, your bone marrow. It takes a massive toll on your energy. So that miserable feeling of having the flu, it isn't just the virus doing damage. It's essentially friendly fire from your own interferon response causing systemic collateral damage to win the war.
To prevent those severe symptoms from doing permanent damage or even killing you, our cells utilize a built-in negative feedback loop. They use suppressors of cytokine signaling, or SOCs, to safely turn off the alarm. As the signaling cascades turn on the antiviral genes, they are simultaneously turning on SOX genes. And as these SOX proteins build up, they physically block the signaling receptors and mark them for the trash bin. The whole system is beautifully designed to automatically hit the brakes once the immediate threat is handled.
But of course, the war never truly ends. Because to ensure their own survival, viruses are constantly evolving counterattacks. Think about it. If a virus cannot bypass our barriers and our antiviral state, it just ceases to exist. Poof, gone. Therefore, every single virus that actually successfully makes us sick carries very specific molecular weapons designed to sabotage our signaling.
Take SARS-CoV-2, for example. It actually evolved a specific protein called ORF7A to antagonize and completely defeat that tetherin trap we looked at just a minute ago. Through sheer evolutionary pressure, the virus developed a tool that binds to tetherin and stops it from anchoring the virus to the cell. It effectively snips the molecular chain, allowing the virus to break free and spread rapidly through the lungs. And honestly, this is a huge part of exactly why SARS-CoV-2 was able to become such a successful and devastating pathogen.
And you know what's wild? This microscopic arms race isn't just happening in humans. Even bacteria have an innate defense system to battle invading viruses, called phages. We always hear about CRISPR as this revolutionary gene editing tool in modern labs, right? But out in nature, it is literally a bacterial immune system. Bacteria capture little fragments of DNA from invading viruses, store them away, and then use them to guide Cas proteins to chop up that exact virus if it ever tries to invade again. And predictably, just like human viruses, phages have evolved their own anti-CRISPR proteins to fight back. It's relentless.
So, as we wrap up this explainer, I want to leave you with this final thought. In this eternal, escalating arms race between invader and host, how will our microscopic defenses evolve next? The viruses are constantly changing the locks, and our cells are constantly forging new keys. Understanding this magnificent, intrinsic, and innate immune system doesn't just help us appreciate our own biology. It's the absolutely critical foundation for designing the next generation of life-saving vaccines and therapeutics. Thanks for joining me on this deep dive into the microscopic battlefield.